Thank you. Thank you very much, and thank you to Matt for organizing this fantastic meeting. I changed a little bit the title of the presentation to capture some of the most recent work that I've been doing in the laboratory. It's a special year for UConn, so if you see some basketball in my introductory slide, you know, it's the second time in history that both men and women wins the double NCAA tournament, so it's a great year for us, you know, so indeed. But I'm going to be talking about science, okay? So there's what I really want to do is to talk about, you know, there's this whole notion of what is adaptation versus an adverse reaction, and in my particular case, I'm going to talk about drug hepatotoxicity and some of the efforts in my laboratory to identify genetic determinants of adaptation, and the way that we do this is by using acetaminophen as a model compound. So I'm going to show you the model that we have developed. But in addition to that, you know, there are basically two models that we have in our laboratory. One, it's where we produce adaptation in response to mild hepatotoxicity. And this goes back to some of the work that Harry Mahendley did about 30 plus years ago, where he shows that if you pre-expose animals with a low to modest hepatotoxic dose of things like carbon tet or thiocyanamide, you wait an adaptation period and then you come in with a subsequent much higher dose that could be lethal, that those mice indeed, or animals, rodents, develop this tolerance or this adaptation. So basically, we're capitalizing on this model that Harry had worked on for a number of years. So I'm going to be referring to the model that we use as autoprotection, particularly with acetaminophen. Harry also showed a different model where he called heteroprotection, in which you could treat animals with, let's say, carbon tetrachloride, and then come back with a challenge using a different hepatotoxicant. And he shows that, in fact, you know, you can have this adaptation when you use two different toxicants for pre and post-treatment, okay? So again, the model that I'm going to be presenting today, it's the acetaminophen autoprotection model that we worked on. Also, in my laboratory, we have been working with peroxisome proliferators as chemicals that confer protection against acetaminophen hepatotoxicity. So this is what I call a category of adaptation where you have adaptation by xenobiotic treatment that doesn't involve that initial hepatic injury. And we talk about those signature toxicological changes that are characterized by hepatomegaly and hypertrophy, mostly dealing with CAR, PXR, PPAR alpha activators. And again, we have used through system biologies and genomics, we have identified a new genetic determinant of acetaminophen hepatotoxicity, a gene called Vanning1 that I won't have the time to talk about, an active collaboration going on with a group of immunologists in Marseille in France. So I don't have to convince you or spend a lot of time showing that, in fact, acetaminophen, acute liver failure to acetaminophen is a serious human health problem. This particular slide here shows the proportion or percentage of cases of acute liver failure in the United States that require transplantation, and you see those numbers are hovering at 50%, which is, to me, really alarming because we're dealing with an over-the-counter medication that is available to a lot of people. This comes from Paul Watkins. It's a paper that he published in Toxicologic Pathology showing the pyramid, even though it refers to idiosyncratic hepatocellular injury, I think it applies to most chemicals that produce hepatotoxicity, in which, you know, a large proportion of folks that are exposed to drugs or chemicals with a hepatotoxicity liability, that they're relatively safe, and then as you go on, you have a subset of patients that they develop increases in ALT, but those patients can actually progress in medication, and they don't develop into further toxicity, and then as you get to the tip of the pyramid, you see that you have folks, a smaller proportion of folks that in fact develop liver injury, and what I want to do is to guide you toward this statement that he has in here, that the possibility that, the intriguing possibility that a single inherited defect in adaptation might be required for a progression of drug-induced liver injury. I would like to challenge this concept and perhaps propose that it takes more than a single inherited defect, possibly multiple defects might be, or genetic polymorphisms might be needed for some of these serious adverse reactions to occur in patients, in individuals. So what I show here is, this is a meta-analysis done by McNeil, which is the maker of Tylenol, where they took 8 clinical trials of patients with osteoarthritis, taking the maximal recommended dose of acetaminophen, which it's 4 grams per day. And there were roughly about 1,500 patients, and what was remarkable, none of all these patients developed a hepatotoxicity, but about 15% of the patients show the following trend. And each graph that we have in here, it's an individual case or individual patient. You see that there's this transient increase in ALT elevation that is certainly below what is considered to be the upper level of normality. And you see that even with continued treatment, these patients, the ALT values, they resolve by themselves without the necessity of discontinuing treatment, indicating that in these patients there's some sort of adaptation to that initial. And in fact, those patients, they came in with no or low ALT values that are considered to be normal. So that transient blip appears to correct itself without the necessity of discontinuing treatment, indicating that there's some sort of adaptation that is taking place in these patients. And basically, our studies are geared toward answering and identifying what are some of the factors dictating this adaptation. Okay? So I'm going to be talking about this model of acetaminophen autoprotection. And in our model, what we basically do is we follow what Harry Mehendley did back then for carbon tetrachloride and thiacetamide. We treated animals with acetaminophen at a dose of 400 mg per kg. We waited 48 hours, and then we came in with a greater, higher dose of acetaminophen, or either 600 or 700 mg per kg. And then 24 hours later, we sacrificed the animals and did analysis of toxicity. And you can clearly see that those mice that are pre-treated with acetaminophen and then challenged with the much higher dose of APAP, those animals, in fact, have a significant reduction in evidence of toxicity by elevations in ALT, and we confirmed this also with histopathological analysis. Initially, we started to look at the potential role of liver transporters, and that's where got me into the field of transporters in the development of this autoprotection by acetaminophen, because we saw that some of the phenotypic features of this APAP autoprotection model resembles somehow what happens in cancer multi-drug resistance. We did a number of studies where we determined that that priming dose of acetaminophen that results in that adaptation does not change the way that the second dose of acetaminophen is metabolized by cytochrome P450 or by detoxification through glucuronide or sulfate conjugation, so that tolerance that these mice develop are not due to simply the way that the second dose of acetaminophen is metabolized. So that to us resembles somehow that multi-drug resistant phenotype that you see in some forms of cancer, in which alteration of transporters is critical for that tolerance that cancer cells develop. And in fact, we did a very comprehensive analysis where we looked at expression and multiple uptake and efflux transporters, the ones that Angela Smith described before, and basically this summarizes the work of Lauren Alexunes, so it's about 75% of her thesis summarized in a single slide. She doesn't like when I trivialize her data into a single slide, but it involved actually a lot of work. So that priming dose of acetaminophen that allowed these animals to develop this tolerance, this is exactly how the liver looks like at the moment that the animals are getting that second dose of acetaminophen. What we see is that the expression of a number of uptake transporters, the OATP family of transporters that Angie talked about, we see that there's a trend for decreasing the expression and function of those transporters. At the same time that when we look at efflux transporters, those involved in moving chemicals from inside of the hepatocyte back into the blood or into the bile, we see that the expression of those transporters increase. And you can clearly see here that MRP4 is listed or illustrated in a different font color because this is the most significantly upregulated transporter that we see with that dose of acetaminophen that leads to this adaptation or this tolerance. The expression of MRP4 is nearly absent in the normal liver, and we see that in response to acetaminophen, we have a very nice centrilobular localization that is very discrete, about 2-3 cell layers away from the central vein. And what is critical, we use carbon tetrachloride, which is a chemical that is highly lipophilic. It gets inside and outside of the liver in a matter that is independent of transporter expression, and we see the same pattern. After our work, there are multiple other hepatotoxicants and liver disease conditions in which MRP4 is certainly induced, indicating that these changes in MRP4 expression is not due to the chemical that is producing the damage, but potentially a generalized response to mild liver injury in our case, and a compensatory response that we feel it's somewhat contributing to that tolerance that these animals develop. Here is a list of some of the chemicals that are transported by MRP4, conjugated steroids, prostanoids, and also cyclic nucleotides. So those are signaling molecules that we're paying close attention to, and we're trying to understand exactly how changes in the expression of this transporter and changes in those signaling molecules might be somewhat related to this development of tolerance that we have here. A number of pharmaceuticals such as methotrexate, conjugated bile salts, uric acid, and acetaminophen sulfate. When we look, we did a study in which we look at the correlation between gene expression for MRP4 and hepatotoxicity, and you see that in this particular study we have a number of mice that receive that same priming dose of acetaminophen that we talked about. We look at gene expression at 12 hours, and it's real typical that when you treat a number of C57 black mice that you get a lot of animal-to-animal variability in toxicity, which is illustrated here by the ALT values, as high as 3,000 in one mouse, and some mice that, believe it or not, actually barely responded to that toxic dose of acetaminophen. And when we correlate the extent of damage to gene expression, you can clearly see that those animals that they have very high expression, gene expression of MRP4 in response to acetaminophen, those are the animals that are very tolerant to toxicity. And on the other hand, you see that those animals with very high ALT values, they have significantly lower level of expression of MRP4. Briefly, one of our goals in our laboratory was to understand signaling pathways and involved in the regulation of MRP4, and we have published a number of papers indicating that both the NERV2 pathway is actually critical for regulating MRP4 in this model of autoprotection that I talked about, and also the presence and activation of Kupffer cells. We also determined that Kupffer cell activation is critical for this regulation of MRP4 that we see in response to acetaminophen under the conditions in which these animals develop that degree of tolerance. We're finishing up a number of studies. Something about MRP4 is that it's one of the ABC transporters that we know relatively little about the structure and the regulation of the promoter. We have finished a number of studies that are now in preparation, where we have characterized the structure and the regulation of the promoter of MRP4. So that's work that we have completed and that we're moving on. And it'll be naive from my part to try to convince you that MRP4 is the only genetic determinant that is responsible for conferring such a profound tolerance to acetaminophen hepatotoxicity. So in collaboration with folks at Pfizer, we decided to do a series of transcriptomic studies to identify other genes that of interest that we think are potentially involved in this protection or tolerance to acetaminophen toxicity. We have essentially the same treatment scheme that I showed you before, pre-treating the mice and then re-exposing the mice to a higher dose of acetaminophen. In this case, in addition to a 24-hour sample collection, we look at samples, tissues earlier at 4 hours after that second re-exposure. And we have the different treatment groups in here at the bottom. Of course, you know, we did AFI chips, you know, to look at genes that are differentially expressed. I'm not going to spend a lot of time going through the differential analysis that we did to basically distill the data to the genes that we feel are genes of interest. But if you look under C, we have 13 genes in common on those two groups of animals that receive acetaminophen as pre-treatment and post-treatment, tissues collected at 4 hours or 24 hours. So these 13 genes are what we call the genes of interest, that we think there might be some genes in there that are of potential importance to this development of tolerance to acetaminophen hepatotoxicity. The genes in black are genes that are downregulated. So out of the 13 genes, most of them were downregulated. I have no idea what some of these genes are. So if your gene of interest or your favorite gene is sitting there, you know, I'll be happy to talk to you. And then we saw 3 genes that were upregulated. The one that really caught our attention is flavin-containing monooxygenase. And the remaining of the talk, I'm going to explain to you how we feel, and we have generated data that indicates that FMO3 is what we believe to be a genetic determinant of acetaminophen hepatotoxicity. For the drug metabolism folks in the audience, this might be interesting because FMO3 is a monooxygenase similar to cytochrome P450, in which produces the same type of metabolites as cytochrome P450, although we see that most of the metabolites that are made I consider to be non-toxic. It is an NADPH-dependent enzyme. What we know is that the expression of FMO3 in the mouse liver is nearly absent. And when we look at females, so there's a big gender difference in expression of FMO3. Until recently, what was fascinating about that finding is that until recently, FMO3 was considered to be non-inducible. Through decades of studies, you know, nobody ever showed that FMO3, in fact, was an inducible gene. Only a recent paper showed that FMO3 can be induced, gene expression of FMO3 by activation of the AHR receptor, okay? And as far as we know, there's no reported evidence indicating that FMO is involved in acetaminophen metabolism. So this is, we did an extensive, a comprehensive time course using that dose of acetaminophen that leads to development of tolerance, and you can clearly see that we have this dramatic increase in gene expression for FMO3. We also see that at the protein level, when you look at animals that are pre-treated and then challenged with acetaminophen, there were no commercial antibodies that would recognize mouse FMO3, so we had to develop our own. we can clearly see that change in gene expression occurs in along with an increase in protein. As you can see here, we have dramatic increase in FMO3 protein expression. And this is also here to illustrate that when you look immunochemically, you see that in male mice, these are naive animals, untreated animals, two different magnifications, essentially no expression of FMO3 in males. And you can clearly see that in female mice, untreated female mice, FMO3, it's significantly expressed in portal regions. So we also know from the literature, and this work was done by Yvonne Wan at Kansas, she's done some work indicating that female mice are highly resistant to acetaminophen hepatotoxicity. And this comes out of Yvonne's work, in which she shows that 18 hours after acetaminophen treatment, you see that males have their typical susceptibility to acetaminophen hepatotoxicity, and the same dose that produces this degree of toxicity, when you look at females, the females are fairly non-responsive to acetaminophen tox. So the question, we ask a simple question: if we inhibit FMO3 expression in those mice, in female mice, does that sensitizes female mice to acetaminophen hepatotoxicity? And in fact, that's the case. Here we have male and female mice receiving, in the case of the males, receiving only acetaminophen. You see that your typical hepatotoxic response by acetaminophen in males, when we inhibit FMO3 with the use of this chemical methimazole, that is a FMO inhibitor, we see that we do not change the hepatotoxicity that males exhibit, so it doesn't affect the degree of toxicity that acetaminophen produces in males. But here we have the females, and normally what happens is females are very resistant to acetaminophen toxicity, and then when we inhibit FMO3 in females with methimazole, you see that females become as susceptible to acetaminophen as males. So we have a proof of concept that by inhibiting FMO3, we sensitize these female mice to acetaminophen tox. Okay? If I can escape from here. Okay, so something else. Let me see if I can get out of here. Whoa. Some people will say that I would like to see myself on vanity. Okay, so by modifying FMO3 activity, you see that we have changed the sensitivity of these female mice to acetaminophen hepatotoxicity. So to finish up, what we're trying to do also is to set up an in vitro system in which we overexpress FMO3 as a way of further studying, the susceptibility of hepatocytes to not only acetaminophen but also other chemicals that produce oxidative stress. So we're employing the HepRG cells, and as some of you know, these HepRG cells are bipotent progenitor cells that were derived from a hepatoma in a patient with cirrhosis that was secondary to HCV. Upon differentiation, you have two morphological distinct populations, hepatocyte-like cells and biliary epithelial cells. And those cells can actually be differentiated with a differentiation cocktail that I'm going to explain briefly here. And those cells actually behave like hepatocytes. They respond to cytochrome P450 inducers, so they are the closest thing to hepatocytes that you can get. So we actually use a lentiviral vector to develop clones overexpressing FMO3, and you can clearly see, we selected 3 clones. We have the empty vector, the low expressing, what we call low expressing FMO3, and high expressing, and we were successful in doing this. And This is basically the way that you do the differentiation in the cells. The cells are grown in what we call the culture media for 14 days. From 14 days to 23 days, they are on a mix of growth and differentiation media on a 1-to-1 ratio. And then from day 23 to 35, so it takes about 35 days to get the cells ready to do a study. They're actually on differentiation media. My PhD student came to me and he said, Jose, there's something really strange here, because what happens is that the cells that are expressing high levels of FMO3 are differentiating even in growth media. I don't have to even put differentiation media for these cells to become differentiated. And what is quite interesting, so these are the empty vector cells going through the different differentiation stages, and you go from undifferentiated cells to differentiated cells. And then when you look at the gene expression level, the low expressing clone and the high expressing clone, you see even early on day 14, these cells are differentiating by themselves without the necessity of putting any differentiation media. So FMO3 evidently is doing something to the cells that is allowing these cells to differentiate even in the absence of this particular protocol that allowed these cells to differentiate. And actually what we did is we send, and they respond to CYP3A4. It's commonly used as a marker of differentiation of the cells, and you can clearly see the difference when you look at the empty vector compared to the high expressing clone and the response to rifampicin. You can clearly see that gene expression is substantially greater on those cells that are overexpressing FMO3 in high levels. So to finish up, in addition to that gene expression analysis that came up with those 13 genes that I mentioned to you in collaboration with Pfizer, we're also doing some system biology approaches where we're identifying pathways from the same sample set, and for that we have used this particular engine that is called the causal reasoning engine. This is a novel, recently developed computational platform that provides a hypothesis or inferences on the upstream molecular events that best explain the observed gene expression profiles that we have by interrogating prior biological knowledge. So what happens is that this will provide you with this hypothesis that can be further integrated into networks and into pathways, okay? So the CRE will also rank this hypothesis based on degree of significance. And I don't know why this is, I know what happened here. The RNA-seq, the slides are, just bear with me for 1 second. RNA-seq is here to illustrate that, in fact, the samples from those cells that overexpress in FMO3, they have been sent for RNA sequencing, so I'm waiting for this data. But nevertheless, you know, our computational engine that I mentioned to you developed this hypothesis that best explain the gene expression data that came out of the array. We have used this to identify new pathways, and in fact, this is what came out from this particular analysis. You can clearly see that those hypotheses are basically composed the rankings of the hypotheses across the different treatment groups. Reds are the hypotheses, you know, that are most significant. It ranks from 1 to 10. The yellow one are hypotheses that ranks from 11 to 25. And then the greens are the hypotheses that are ranked over 25. And you can clearly see here that some of the hypotheses for this autoprotection model, out of this computational approach, you know, we have a lot of things related to immune response. In fact, response to hypoxia ranked #1 for all the treatment groups. You see things like IL-1 beta, IL-1/6, NERV2, and and I believe there are a couple of other cytokines in here that came up as significant hypotheses associated with this model of acetaminophen protection, and this is something that we're clearly following up in details. And also what I want to show is that this computational platform also provides you what is called a mesh cloud, and the mesh cloud, what you have is these terms, and the size of the font of the term tells you the significance of that particular term. And for example, this is the hypothesis for NERV2 in one of the treatment groups, acetaminophen, pre-treatment, post-treatment at 4 hours. And then this is vehicle followed by acetaminophen treatment at 4 hours. And then these are the changes in genes that are supporting that induction in NERV2 for both treatment groups, and then the terms and the size of the font indicates the significance of this particular term. In this case, you see metabolic detoxification, oxidative stress, which clearly matches, a hypothesis consistent with NERV2 response in these tissues. Okay? So as a summary, I showed you data where we have a comprehensive characterization on the expression of ABCC4 MRP4 transporter, transcriptomic analysis that help us identify flavin-containing monoxygenase 3 as a differentially expressed gene that appears to have a role in dictating susceptibility to acetaminophen hepatotoxicity, particularly from those studies where we inhibited FMO3 in female and male animals, and we see that the susceptibility only changed in female males. I showed you a unique phenotype of those FMO3 overexpressing cells in these HepRG cells in which they allow to differentiate in conditions that they should not be differentiated. And then I show you some of the approaches that we have used to identify pathways and to put those pathways in some sort of biological context for the different treatment groups that we have in here. Okay? So with that, I would like to thank some of the people at the University of Connecticut that did all the work, some of my collaborators at Rutgers University, Kirk Klass and Angie's here, Nathan, long-term collaborators, and funding agencies that make this work possible. And with that, I'll be happy to answer any questions. Yes?
Have you looked in GCLR knockout mice to see if your auto protection goes away?
No, I haven't. That's something that I haven't done.
I mean, I wonder how much of it is just mediated through glutathione sensing. Because NERV2 mice are very sensitive to AT.
Very sensitive.
But I expect that they don't have the auto protection. Have you looked at that?
We have inconclusive studies. We have done one study in which there appears to be that there's still some degree of protection. Yeah. Well, those are studies that we have to review. But the results are inconclusive at this moment. Yes. Go ahead.
I assume you're thinking that acetaminophen is a substrate for HepatiM3. Have any thoughts of what it's doing?
I've spoken to Ron Nice. I engaged Ron as a collaborator in this work. So we haven't done any work to try to study and to understand if there's any catalytic activity in the presence of acetaminophen. Is something that we certainly have to do. The issue is that when you look at the time frame for induction of FMO3, it's happening very late, way after acetaminophen is gone from the system. So the question is, is it really critical for the metabolism of acetaminophen itself? So.
Yeah, we've reported some studies that are somewhat similar to the paradigm that you're investigating. Specifically, what we've shown is that acetaminophen will protect against the carcinogenicity of several aromatic amines. And the only explanation that we've been able to come up with for this effect is that in these studies, acetaminophen reduced the induced cell proliferation there anything in your gene data that would relate to an effect like that?
On cell proliferation?
Yes,
To increase cell proliferation, you mean?
decrease.
Actually, we feel that is the opposite, you know, because we have some data indicating that we have done some work where we pre-treat the animals with acetaminophen, and then we give the challenge dose. At the same time that we give this challenge dose of acetaminophen, we treat the animals with colchicine to prevent cell proliferation, and we see that those animals that are receiving colchicine, along with that second dose of acetaminophen, they lose that resistance, you know. So we think that is the opposite, that cell proliferation is, and it goes along with what Harry Mehendel has suggested, that compensatory cell proliferation is actually critical and somewhat needed for that tolerance that these animals develop. I'll be happy to discuss that with you, but we have addressed that.
But one of your genes that was down was one of the ribonucleotide reductases, and so if that's critical for the proliferation, that could explain it.
I'll take one more dance because we really need to move. Yeah, go ahead. No.
Is there any impact between the FMO3 expression and R34 expression in this?
That's something that we're doing. We're doing alignments. We're looking at the promoter of both genes, you know, to see what type of responsive elements or regulatory elements might be common to the two genes, you know, to give us an idea whether there's a common universal signaling event that might be critical for the regulation of both genes.
Could it be simple as FMO3 producing some metabolite
Possibly.
that is acting as a
Possibly. We're looking into that possibility too. That's actually, we feel that maybe it's metabolizing or generating an endogenous substance that is certainly playing a role in the. Yeah.
Thank you.
Sorry, Nathan.